Here today. That's great. So I'm going to talk to you about Evo2, our new DNA language model. So as we all know, biology is encoded in DNA. And from the central dogma, we have information flowing up from DNA. And whether a interaction happens at the level of regulation, whether it happens at the level of RNA or at the level of protein, that information is ultimately passed down from generation to generation in the DNA. To us, however, DNA is a foreign language. So when you ask to be or not to be, you can fill in the blank. But when you ask ATG, it's much harder. And what comes next is probably a repeat. So language models can learn language from data. Human language was foreign to transformers before pre-training. And DNA is also foreign to language models before pre-training. By pre-training on a simple task like next word prediction, you can imagine that this actually imbues information about complex associations and even about the world itself. So through learning next token prediction in human language, you learn something about the rules of grammar, about semantics, about the world, about sentiment, and so on. These are just a few examples. But hopefully you can see how if you want to be able to predict just the next token or the next word correctly, a model that does that well would be able to learn something about these complex tasks. So we're not talking about human language, we're talking about DNA. And in biology, through the process of evolution, information from DNA flows up through the central dogma through various mechanisms to create organisms' phenotypes. And then selection, natural selection, acts on those phenotypes, changing what DNA is passed on to future generations. So as as evolution acts on the phenotype, it changes the genotype. And we hope that by learning on the DNA, we can learn some of this complex biological mechanisms that are reflected in and captured in the sequence variation that is observed in the DNA. So a simple example might be an interaction between two different genes that may be far apart in DNA sequence, but in physical space, these two genes interact and they have some type of amino acid binding between those two genes. And if one of them mutates, that binding may be ablated. So over evolutionary time, you expect there to be covariation between these different parts of the DNA. And so this is just one example of the type of mechanism that you'd expect to be able to discover in sequence variation. And by training language models on sequences, you hope to learn something about these kinds of relationships. And just like next token prediction of human language may help us create a model useful for many things in natural language processing, we hope that simple tasks like next token prediction in DNA can help teach us useful things from disease genetics to protein biochemistry to RNA structure to even interactions between multiple genes in complex systems. Evolutionary signals, like the ones I described, already power the best predictive models of proteins. So AlphaFold famously uses a multiple sequence alignment input, which is crucial for its high performance. And this was shown in follow-up studies that the initial guess provided by the covariation in the multiple sequence alignment and in the Evo former block is crucial for then subsequent refinement, which is handled by the structure module. And similar analysis suggests that in language models which operate only on single sequences of proteins, like ESM2, you can still see the same behaviors in terms of failure modes as statistical models of sequence evolution. So the idea is that these evolutionary signals are very useful. They help power our best predictive models, and we want to be able to learn them from DNA. So we have great models of protein sequence. How can we learn from DNA directly to capture different scales and modalities? So in biology, we care about a lot of different scales, from single elements that are just tens or hundreds of nucleotides long to entire genomes and the complex interactions that they capture. So we want a model that may be able to reason over all of these. Genomes are also uniquely hard to model, not just because of their scale, but also due to their composition. Genomes have a lot of repeats, both simple and complex, and these make up a large fraction of many genomes. So selfish genes that copy themselves, repeat expansions, simple repeats versus complex repeats, all of these will be interspaced with essential genes with important biological functions. So learning from DNA exposes you to these challenges that learning from proteins may help sidestep. It can take thousands of these DNA base pairs to encode just a single gene, but any one of those base pairs may be essential, and a mutation in it can alter the function or even completely ablate it. So we need a model that is able to reason at scale, but still pay attention to small changes in DNA sequence. So we developed EVO2, a long context DNA language model trained across all domains of life in order to learn DNA from the small to the big scales. In order to do this, we first developed striped hyena 2, a architecture specifically designed to improve throughput and quality when modeling DNA sequence. So compared to EVO1, which uses striped hyena 2 is a multi-hybrid combining 3 different types of convolutional layers with transformer layers. So these new types of convolutional layers are the short and medium convolutions. These are similar. These use a similar mechanism to a classic CNN in order to mix information across positions. And so these are more efficient than long convolutions or transformers. And we find that they're well suited for modeling DNA, where there's, of course, a lot of important local motifs, as well as important long-range interactions. So this improves throughput substantially, especially at long sequence length. One of the goals was to consume large numbers of tokens at long sequence length, which is challenging, especially with transformers architectures. So by developing a more efficient architecture, we were able to significantly improve throughput at context lengths that we cared about. And details about the algorithms and systems work that enabled efficient training is available in our companion preprint, which I suggest that you check out if you're interested. So we have an architecture that trains efficiently, and we found in various controlled experiments that it has low, potentially even better perplexity compared to other architectures. So now we need a data set. And so to do this, we collected 9 trillion base pairs across all domains of life by combining various publicly available data sets and applying deduplication methods in order to balance the quantity of data with its diversity, avoiding the overrepresentation of very commonly sequenced species, which would happen if you used all the data. We find that data composition and augmentation is critical for training on DNA. So in part due to the issues about repeats that I mentioned earlier, and also in part due to the redundancy and similarity between many genomes, we conducted different experiments and found that for downstream tasks, how you adjust the data composition changes performance significantly. So we specifically looked at performance on zero-shot evaluations of disease variants in humans. And saw that adjusting the training composition could dramatically change the performance on this. So we've decided to focus heavily on these eukaryotic genic regions instead of on whole eukaryotic genomes. So these genic regions are data augmentations that we apply during the pre-training phase of the model, which is the majority of training time. And we focus on the mRNAs as well as the promoters and splice sites. And then we also focus on these 5 kb windows where we're taking 5,000 base pairs on each side of the exon and joining it in order to focus on genes and on proximal non-coding DNA. And so this we find improves performance both on coding, but especially on non-coding sequence and proximal DNA. So as alluded to earlier, EVO2 is trained actually in 2 phases, a pre-training phase, which is at 8,000 context length, and a mid-training context extension. And so those 2 data compositions earlier are altered, where during the first phase, we hope to learn functional elements. And then during the context extension phase, we hope to learn how these functional elements relate to each other in longer sequence. And so we adjust the data composition accordingly in order to first focus on those genic regions that I mentioned earlier, and then during context extension to focus on whole genomes, while maintaining some percent mix of short sequences. So we find that as we scale the model and context length, the loss continues improving, suggesting that the model is taking advantage of its scale and its context length. We then perform quick tests at each stage of context extension in order to make sure that we're using the context length efficiently. And we find that the mutations in 2 homologous parts of DNA spread out throughout a random background sequence are connected to each other by the model. So changes in one region that is homologous to another change the model's predictions for that second sequence, even at very long context lengths, up to the 1 million base pair length that we trained at. So validating that the model is able to use this context to adjust its predictions. So now we've pre-trained a model and we feel like it's able to improve its loss and is able to pass this simple needle in the haystack test. We then wanted to look at how the zero-shot likelihoods reflect function across DNA, RNA, and protein. One of the exciting avenues of DNA language models is providing a unified likelihood score for mutations, whether they are coding or non-coding. And so you could imagine in protein engineering or engineering of complex systems where there's multiple interacting pieces, or in disease variant prediction, you care about not just the fitness of mutations of one type, but you care about different types of mutations and how their likelihoods relate to each other. And so what we're doing first is seeing if this pre-trained model, where it's just trained on a bunch of sequences, if the likelihoods that come out of it are calibrated and are predictive of fitness across these different modalities. So one thing to check is whether the mutations match expected biological pieces of genes. So when we introduce mutations along the sequence around genes and their start sites, we can see that EVO2's likelihoods of the mutations match the codon pattern. So the gene start is conserved, and then for the subsequent pieces, the wobble-based third position in the codon is less conserved. And so this suggests that the model has learned the relative importance of gene starts and the relative importance of the first 2 positions in codons. And there's also some signal around the 5' UTR for prokaryotes, and then less of that signal in eukaryotes, as you might expect. So now that we've validated it against a kind of sense check, we want to then look at data from essentiality and mutagenesis studies. So we correlate EVO2 likelihoods with the measured gene essentiality in prokaryotes, and we find that EVO2 is able to predict the essentiality of various genes for phage and bacteria actually better than EVO1, despite having been trained on all domains of life instead of just prokaryotes. We then do a similar analysis for a human long non-coding RNA dataset, where the essentiality of these long non-coding RNA genes was measured in human cell lines. And we see once again that the likelihoods coming out of EVO and how they change when a mutation is introduced correlates with the essentiality of that long non-coding RNA gene in cell lines. So mutations seen as less likely by EVO in long non-coding RNA seen as more essential by EVO match the experimental readout. And so this is especially exciting because it's a non-coding RNA, something that you can't score the likelihoods of using a protein language model. So now, having compared with different mutagenesis datasets, we also wanted to look at human disease variants. Here, similarly, we're looking at the likelihood of the mutation, but since we're comparing mutations in many different parts of the sequence of the human genome, we have to normalize to the wild type sequence. So what we're looking at is the delta between the mutation sequence and the wild type sequence at that position, allowing us to compare for many different mutations. So we find that EVO2 is able to predict substitutions and non-SNVs for both coding and non-coding mutations. So for coding mutations, EVO2 is competitive, although somewhat worse than the best models. But for non-SNVs, which are short insertions and deletions, EVO is state of the art. And then for non-coding, EVO does very well, including for both the substitutions and the indels. So here we see that EVO, through the pre-training on DNA sequence, is able to create this zero-shot likelihood that correlates with human disease variants, whether they are coding or non-coding, and whether they are substitutions or indels. We then repeat this analysis looking at a BRCA1 allele, which has a deep mutational scanning data set. And so it's the same analysis, but now we're classifying the loss of function variants in the BRCA1 deep mutational scanning experiment. And so for coding, once again, EVO is not as good as the best specialized models, but it is state of the art for non-coding. And when putting the variants together, EVO also maintains performance and is state of the art. So it's important not just to be able to predict coding or non-coding variants separately, but also to have the calibrated likelihood when they're combined. And just like you would expect, fine-tuning models on top of EVO2 can improve prediction accuracy. So if we create a train set, train a small model on top of the EVO2 embeddings, you can drastically improve the performance on a test set. And so similarly to approaches that were done using embeddings for protein language models, we hope that EVO2 can empower better predictions by providing these good zero-shot scores, but also these embeddings that can then be trained on top of. So EVO's learned something, right? It's pre-trained on all of this trillions of DNA bases, and it's creating likelihoods that match with deep mutational scans and that can predict human disease variants. But how is it doing that? What is it actually learning? And to try to start addressing this, we turn to mechanistic interpretability, a method developed to interpret large language models. So you may have seen the example from Anthropic, where they were able to identify features in large language models that correspond to specific human concepts. So what this means is that there will be a specific neuron or set of neurons in the large language model that matches to a concept, a coherent single, a concept. So this example being the Golden Gate Bridge, where Anthropic found a concept, a feature that matches with our human concept of the Golden Gate Bridge. And whenever the Golden Gate Bridge is mentioned in text, this fires. And then you can actually up or down weigh this Golden Gate Bridge feature in order to alter the behavior of the Claude model. So we want to find similar features in Evo2. So to do this, we team up with Goodfire and trained sparse autoencoders on Evo2, and then use these biological features to try to annotate and look at genomes and interpret what the model has learned. We find that Evo2 has learned protein, RNA, and DNA level features, as we would hope. We do this by looking at the activations of different features from the SAEs, and then matching the activations of those features with annotations that you have for different features. So by doing this across large numbers of features, we're able to find which SAE features match our annotated concepts, if any. So Evo has learned RNA and protein level features with specific features that fire, for example, on beta sheets and alpha helices, and then other features that fire on RNA. So despite having been trained on unannotated DNA sequence, Evo has some set of neurons that are very specific to these higher level biochemical features like a beta sheet, or to these biological features like a tRNA. So this is encouraging. It suggests that the models are learning these useful concepts and rediscovering things that scientists have discovered just from training on raw DNA. We find similar features for DNA regulatory motifs. So some Evo features specific to different transcription factor family motifs. And we also find higher level features like a phage-associated feature that fires on prophages, CRISPR arrays, and even individual phage-derived genes. So this is exciting because a phage is quite a complicated concept. It's almost life, and there's many pieces. And the fact that you're learning a single feature that represents this suggests that pre-training on DNA is picking up some of these interesting higher level biological concepts. And importantly, this is true for both eukaryotes and prokaryotes, where you're learning features for both, and often features that may even generalize. So here there's a set of features we found that are specific to various various parts of genome architecture. So the starts of genes and the introns and coding sequences. One very interesting result from large language model Mechanistic was the feature for bugs. So there was a specific feature from Anthropic that would identify bugs in computer code. And actually, we were able to find a feature in Evo that identified bugs in genetic code, specifically frame shifts. So this feature fired on frame shifts, whether they were in whatever various different genes. It was specifically firing on frame shifts, and with a weak firing on some other types of bugs like premature stop codons. So there's actually a visualizer available on the ARC website where you can look at the activations of these features in different sequences and play around with that. So we've explored Evo2's features, and we found encouraging signs of these higher level biological concepts. Now we want to use this generative model to generate. So Evo2 can be used to generate just like GPT. And in this case, we look at genome generation from prompting, looking at examples from mitochondrial, prokaryotic, and eukaryotic generation. So we start with the human mitochondria, which has a 16 kb genome. We prompt with the beginning of the mitochondrial genome and generate a fixed number of tokens, and then compare these generations, annotate them using mitochondrial annotators. We find that the Evo-generated mitochondria maintain the ordering of genes and mostly maintain which genes are present. However, there is significant sequence level diversity among these genes. So the model is maintaining some concepts on the synteny of the mitochondrial genomes, but diversifying their specifics, perhaps introducing additional genes or reshuffling genes, splitting genes into two. When we co-fold together genes from one single mitochondrial generation from Evo, we're able to see that the structure of the complexes match those of wild-type sequence, and there's some amino acid sequence diversity around 80 to 90% for most of these proteins. But keep in mind, these are highly conserved proteins, and so the divergence of some of these mitochondrial proteins from humans to fish may be 80% amino acid identity. So this is a very high amount of diversity for this very highly conserved set of mitochondrial proteins. So Evo is able to generate these proteins, this mitochondrial genome, and then on that genome, there are these pieces that then co-fold together in AlphaFold into the shape that we expect, and they offer some variable amount of diversity. So this is exciting as a showcase of generating a multi-gene complex system. We then look at bacterial genome scale generation, prompting with the M genitalium genome, which is 600,000, the whole genome is 600,000 base pairs. So you prompt with 10,000 base pairs, and we generate 600 base pairs from that prompt. We then annotate using Prodigal and compare and analyze the annotated genes for homology with real genes. And so we see that Evo2 has significantly improved over Evo1 in terms of generating many more genes that are homologous to some real gene. And the distribution of the lengths of genes and their composition also looks significantly better than Evo1. So we can look at the fraction of genes with a homology hit to PFAM, and here over 60% of the Evo2 genes have that compared to less than 20% of Evo1. And then we can also see that the lengths of the genes that Evo generated are matching the distribution of the natural M genitalium genome, and that the secondary structures of those genes, when we fold them and compare the secondary structure distribution, it also looks reasonably similar to the wild type sequence. So next, we looked at yeast chromosome generation, prompting with a piece, the start of yeast chromosome 3, and then annotating using GeneMark and doing, again, a homology analysis. So here, again, Evo2 is able to generate yeast chromosomes that have annotated genes, introns, promoters, and tRNAs. And then looking at the distribution in terms of genes, it's a little bit lower. It's lower than wild type, but then introns looks similar to wild type, and we have significantly fewer tRNAs. There's only about 2 on average compared to 10 in wild type. However, the number of promoters generated seems correct. And then looking at the secondary structure, it looks realistic comparing the Evo-generated proteins to the wild-type genes. So together, these suggest that Evo2 is improving its ability to generate realistic genomes, although there's still room for improvement before you can one-shot generate an entire chromosome and expect it to work. So one way to then build on top of a model that doesn't always work 100% in its single generation is through search and inference-time compute. This is a trend common in natural language processing currently to do more sampling and to do search or reasoning in order to improve your generation. So the model doesn't have to get it right the first time, but you can improve it. And so one task that we applied this concept to was a design task of chromatin accessibility. If you want to encode a desired peak or pattern into chromatin and its openness, you can use Evo as the generative model and combine this with a classifier model in the form of, for example, Informer. And then you classify using the Informer model, and that helps guide your generation towards the pattern you want. So you just keep sampling until you meet your constraints or goals for that sequence. And so here it's running along and generating guided by these design peaks. So in the white bars, the large ones, those are the peaks that are our design goals. And then the blue lines that are being filled in are the generated DNA sequence and how it scores according to the scoring oracle. So by searching over various Evo-generated sequences and then choosing one that is active in your peak, you're able to guide Evo by increasing the amount of sampling you do. So the more you sample, the more you find sequences that match your design goals. So generally, we think that this is an important paradigm to explore where we're combining this generative model that gives us a probability of a DNA sequence with different reward or scoring functions. So by combining Evo with an epigenome predictor like Informer and Borzoi, we're able to guide Evo towards desired epigenetic peaks. You can imagine that building on this paradigm will allow you to generate various interesting biological sequences. So hopefully I've convinced you that by retraining on just DNA, Evo has learned interesting general capabilities, from predicting variant effects by comparing the likelihoods of different mutations, whether they are encoding or non-coding DNA, to discovering biological concepts from just pre-training on DNA, and these concepts then matching our human understanding of biology, to being able to generate realistic DNA from prompts, whether those are organelle, prokaryotic, or eukaryotic DNA. We hope that Evo can serve as a foundation layer that empowers various applications in biology, from molecular applications to specific modalities to larger scale organismal predictions or even this genome scale generation. So read our preprint and check out our GitHub. All of the code, weights, and data for pre-training are open source and available online. This was a huge collaborative effort between many institutions. It's a great team project with many co-authors that worked really hard on this project. And I'm really grateful that we were able to pull it off. And we're also especially grateful to NVIDIA for making this possible and providing the compute needed to train such a large model.